talk with the researchers at SEBA. In fact, thank you for the kind words, and I treat, in fact, Professor Suba as my sister, so I call her sis every time. So of course, it is much more than friends already. And well, thank you for the kind words, Professor Suba. And let me just share with you the topics of my research over the last 2 decades, I would say that, because I started working on this particular protein, which we call heat shock proteins, 20 years back during my PhD in Belgium. So I did my PhD at Ghent University in Ghent, of course, in Belgium. And at that time, this topic is quite new, especially for application in aquatic organisms or aquaculture. So let me share with you some of the basic of heat shock proteins, and thereafter the research that we have conducted over the last 2 decades. And that brings out, I would say, a good story to tell in the aspect of application of heat shock proteins for disease control in aquatic organisms, and particularly aquatic organisms that is useful and valuable for aquaculture. So this type of work is not done only by myself, but a whole group of scientists around the world. And of course, I'd like to compliment several people here who works very closely with me. Of course, Professor Patrick Soglus and Professor Peter Bozier. They are my supervisors. And of course, if you look here, Professor Thomas McRae, the late Professor Thomas McRae, my another supervisor from Canada, and people like Mimi and also Shitao, Tan Jiabao, Chen. All those are postgraduate students who really contributed to the outcome of what we got until today. So when people ask me which type of aquatic organisms do I look into, in fact, I would say that I'm a Artemia man. So if you know brine shrimp, it's called Artemia. So I work a lot of my experiments using Artemia as the model organism. And thereafter, I switch it to Penis vaname, which is the white leg shrimp. So let me just start off. Next, please, Nadira. Let me just start off with a brief introduction about heat shock proteins. Now, heat shock proteins is a group of proteins that occur constitutively in all living cells. And name it, in all organisms, from bacteria, mammals, and all of these living organisms to date possess heat shock proteins. And these type of proteins occur in the cell, and when an organism or a cell is exposed to significant level of stress, particularly heat stress, these type of proteins will be induced significantly from 2-fold to 4-fold or even 5-fold in those cells upon heat shock or any other abiotic or biotic stresses. So next, please. So next, please. Sorry. So what are the primary function of heat shock proteins? They are called protein chaperones. So they are small molecular proteins that is induced, expressed, and assist in the repair of damaged proteins or proteins that are aggregated, that is not in a proper conformation. So those small heat shock proteins would assist in the repairing of this protein back to their proper conformation. And if one of the proteins or a group of proteins that cannot be repaired anymore, then heat shock proteins might assist these proteins, bring them out from the cell for degradation. So they are protein chaperones and of course largely induced in response to stress. Next, please. So there are many classes of heat shock proteins, and if you look at here, we have 10 kilodaltons to 40 kilodaltons. They are categorized as small heat shock proteins, and you have different classes of 60, 70, 90, and 100, and those are bigger molecules of heat shock proteins, and they are categorized based on their molecular sequences. So it's how heavy would be the structure of the proteins. And of course, the naming or the categorization of these heat shock proteins is somewhat different from prokaryotic animal and also in response to eukaryotic animals. So the naming is different, but of course, the molecular structure is quite similar. For example, bacterial heat shock proteins, they are quite conserved and appearing as 60% to 70% similar in terms of the molecular sequence when we compare it to eukaryotic organism. Next, please. So initially, back in, I think, 2 decades ago, people found that when heat shock proteins is expressed in an organism, then those organisms, and particularly those aquatic ones, well, when I say aquatic ones, means it includes fish, shrimp, bivalves, and you name it. A lot of publications have surfaced indicating that when a class of heat shock proteins is upregulated, those aquatic organisms are protected against heat shock, or I would say a heat that usually kills them. So that phenomena is called induced thermotolerance. Let me just give you one example. If we put a fish from 28 degrees C to 42 degrees Celsius or even higher, or 45 degrees Celsius, for 1 or 2 hours, they might die if we impose an acute exposure of heat to those organisms. But if we would prime this organism to a non-lethal heat shock first, let's say when we expose them from 28 degrees Celsius to a very short-term heat shock at 37 degrees Celsius, a temperature that would not kill them, but enough to induce heat shock proteins accumulation in those organisms, then they might survive when we expose them to a temperature that normally kills them, like 45 or even higher. But of course, for your information, the expression of heat shock proteins is usually transient in higher level organism. So when expression is transient, meaning that they can tolerate that stress for a particular time only, and when you prolong the stress exposure time, they might die as well. So this is one of the initial finding indicating that yes, when heat shock protein accumulation increases in an organism, then the induced thermotolerance is also being enhanced. Next, please. And remarkably, that this phenomenon does not only restrict to aquatic organisms, people also see that in plants as well. And not only in terms of heat stress, but tolerant against high salinity, tolerant against desiccation, and many more other abiotic stresses. Next, please. Now, apart from their primary function, being molecular chaperones being assisting in protein damage repair, heat shock proteins has also been well recognized as one of the immune activation factor in mammalian models. So not so much on non-mammalian models, but if you look at a cell which consists of upregulation of heat shock protein synthesis, they are good mediators of sending danger signals to the immune cells for immune activation. And a lot of publications have indicated that they play significant role in toll-like receptors pathway and so forth. So where the main function of those pathway is to transduce inflammatory danger signals to immune cells and activate those immune cells to kill off pathogens. And next, please. And if we look at one of the main model, macrophage, when a bacterial heat shock proteins is attached to a macrophage and through a signaling receptor, many antimicrobial substances might be activated and produced by those macrophages to suppress infection. And you can name it nitric oxide, cytokine, interleukin, and so forth. Next, please. There comes the history, how we started our findings and experiments. Well, I would say 15 years ago or even more, when I was given a topic at that time very challenging to see whether when we heat shock an organism, particularly I work on the brine shrimp, on Artemia, would these animals be more tolerant to a Vibrio challenge? All right. So we started optimizing the heat shock temperature, which means that we impose a short-term non-lethal heat shock, which does not kill the Artemia, but significant enough to upregulate HSP 70 accumulation in the brine stream. Next, please. And next, please. Yeah, at that time, we heat shock them at from 28 degrees Celsius to 32, 37, and 40. And what we saw at that time is by using very simple molecular methods of protein extraction, and after that, with a good antibody for for Western blot probing, we saw that the heat shock protein accumulation, especially HSP 70, is significantly upregulated upon heat shock. And thereafter, next please, we could see that the Vibrio colonization in this Artemia is significantly reduced. And in line with that, we see a twofold increase in the protection of this Artemia against Vibrio Campbelli challenge. So this is one of the major findings that we saw, but we actually do not know what is the reason behind whether the heat significantly reduced the Vibrios by killing them, or in fact they upregulate the immune system of brine shrimp in order for pathogen of Vibrio attenuation. Next, please. So you see that there is a significant reduction. Next, please. And at that time also with my colleagues from, he's working in Sweden now, he found that those protection at that time is due to the enhanced prophenol oxidase activity in the brine shrimp, but no substantial evidence from that time And therefore, we, after obtaining those interesting results, we venture into looking at only heat shock protein 70 by available molecular tools, which I'm going to show with to all of you later. Next, please. So this is the tool that we developed over the last 15 years, and very nicely that after 15 years, we know somehow what is the function or the functional role of heat shock proteins in the brine shrimp Artemia. So we employ RNA interference technology, where this technology was, I think, quite new at that time, 15 years ago, but it's not new anymore now, that we have to undergo a very standard protocol, a lot of optimization. Next, please. So basically, we would like to know the function of heat shock proteins in Artemia, particularly whether this class of heat shock proteins plays a significant role in the immune response and also in the tolerance against Vibrio. And as I said already, this is a very standard molecular technique whereby you can sort of like knock down particular genes in an animal to study the functional role. So by employing RNA interference, of course, you can bring down significantly a class of protein of interest that you want to see. And if you bring down that particular protein, is usually very specific. So meaning that if the RNAi strategy is efficient enough, then you might have a knockoff. And when it's knockoff, meaning that the proteins is not being expressed anymore in those organisms. When you knock down, probably there are some still being expressed, but at a significantly low level. So we have a difference in terms of knockoff and also in terms of knockdown. And knockoff a gene might probably bring it to the next generation. So Nopli or the animals or the babies that came out from the mother might also consist no or even low expression of those proteins, but knockdown usually is on in that generation. All right, and sometimes we do see knockdown that is transient and not at a prolonged duration of time. Next, please. So we started to investigate and see, because we have to produce knockdown animals, HSP 70 knockdown animals, and we have to really go into seeing when we knock down a protein in brine shrimp, what would be the effect on the embryo development of brine shrimp. If the embryo cannot develop, then we do not have that organism for any studies later on. So we have to make sure that the embryo and also the animals grow so that we have the animals at eventually. And of course, we would like to see, because heat shock proteins is very well related to thermal tolerance, we want to see whether if we knock down only heat shock protein 70, would they be good enough to withstand thermal stress. And of course, the ultimate thing that we want to see would be the Vibrio tolerance. Next, please. So first, we look into the sequence of Artemia franciscana heat shock proteins. Yeah, sorry for the figure. I think we don't really need to go into the detail of this, but sequences of Artemia franciscana heat shock proteins is available, so we can get them quite easily. And interestingly, next please, next slide. When we compare the sequences of heat shock proteins of Artemia with other organisms, you can see that it is very, very conserved, highly conserved, and they are sorry.
มีหนังแดงแล้วจะกินหนังส้วงมีหนังส้วงแล้วจะกินหนังแดงแล้วกูไม่แรงแต่กูทำมึงม่วงที่กูไม่ม่วงแต่กูยังมีแรงว่าหัวแดงแล้วก็ทำมือล่วงเอามึงไปคั่วหรือไงพี่แกง
Sorry.
Can off the remote?
มีหนังแดง
If
แล้ว
not,
จะกินหนังส้วงมีห
can
นัง
you
ส้วง
off?
แล้วจะกินแดง
Yeah.
Yeah. Okay. Thank you very much. And
Sorry,
and
Yong.
yeah, no problem. In fact, when we look at the protein sequences or compare those protein sequences with other organisms, you can see that it is they possess very high similarity with almost 80% in terms of identity in terms of the protein sequence. So it's highly conserved. And next, please. So what we actually did was to first construct a plasmid. You can do this very easily now, and gene insertion into a plasmid just can do it with a commercial kit now available easily from the market. And next, please. And when we construct those plasmids, the ultimate goal is to produce double-stranded RNA. So again, this is easily done with also kit nowadays. So very easily that you can produce double-stranded RNA, and we use the megascript RNAi kits from mBion. Next, please. And this is the hard part. In fact, how do you bring those double-stranded RNA into an organism? So when you work with a bigger size of organism like fish, it's easy to bring those double-stranded RNA into into the organism. Just give them an injection, and you can easily do that. But with brine shrimp, which has a size of only 1 centimeter.
I think this. Please mute your microphone, please.
I'm not sure. Is that?
I don't know who is this.
Yeah. It's written not. Yeah.
Not.
Yeah.
Yeah. So not is now been removed from the meeting because
yeah. Yeah. Thank you very much. Thank you. All right. So the idea of bringing the double-stranded RNA into a very small organism is quite difficult at that time. But we were very lucky because we had a very good collaborator, the late Professor Thomas McRae from Dalhousie University. He suggested us to use micro-injection into bringing this double-stranded RNA into the female rootstock of brine shrimp. So we have to get appropriate training at Dalhousie University in Canada. I personally stayed there for a few weeks to look into how we bring those double-stranded RNA into female Artemia. And if you look at the figure here, this is a female Artemia with a fertile uterus. So you see eggs in those broodstock. And what we actually did was to micro-inject the double-stranded RNA with a dye into the uterus, where we can see either nopli or either eggs has been formed in those female Artemia. And once you inject them, and of course, another challenge in the optimization is that how much of double-stranded RNA should we inject in order to get a good knockdown of those proteins. So this has taken a while, in fact, that we keep optimizing the most appropriate concentration, and not only the concentration, but the amount of double-stranded RNA, I mean, in terms of volume, you cannot inject more volume into it, otherwise it is spilled or not all those volume can be injected into the Artemia. So we play around with the volume, and eventually it was found that 250 nanoliter of those double-stranded RNA consisting 80 nanogram of double-stranded RNA is the is the appropriate way of knocking down those protein in the brine shrimp Artemia. Next, please. So upon injection, and of course, let me just show you, this is just a normal micro injector installed on a good microscope. Next, please. And I like to always compliment my student previously, Mimi. She has graduated and now it's Dr. Mimi. She is a lecturer at UMT now and working on similar topics also. So, but she really spent a lot of time in the optimization work. And upon micro injection, next please. This is the animals that you see. And another PhD student of mine, if you look at all those well plates, those pairing is done in these plates where we usually put a pair of Artemia, and upon injection to let them mate, and thereafter to collect cysts and also to collect nopli that is produced by the pairs. Next, please. Yeah. So this is how it looks like after mating. You have the fusion of egg sacs, and thereafter the female will produce nopli and also cysts. Next, please. Yep. And if you look at the nopli, it is only about 200 microns. It's very small, and cysts is even smaller than the nopli, and it is quite delicate to collect them. But we collected sufficient amount of those cysts and nopli, and we have to verify whether those cysts and nopli consist of heat shock proteins or not, to indicate a successful heat shock protein 70 knockdown. Next, please. Yep. And with appropriate molecular technique like real-time PCR and Western blot, we really see that to be on the detectable side of those Western blot, we don't see any more bands as compared to the controls. But of course, we cannot say that total knockoff has been successful. So it is better for us to use the terminology of knockdown. All right, next, please. And one of the interesting results that we saw is that heat shock protein 70 knockdown does not affect the embryo development, meaning that by removing those heat shock proteins, one class of heat shock proteins 70, the nopli still grow to adults. All right, and next, please. But the tolerance against heat is much lower. And also, next, please. And the tolerance when you give them a Vibrio Campbelli challenge also has been reduced significantly. Of course, when we look at the graph on the right side, you can see that when we impose a Vibrio Campbelli challenge at 10 to the power of 8 cells per mil, the tolerance will reduce approximately 30 to 40% of what is being achieved with the control Artemia. And from here, we really think that heat shock protein 70 is required for the protection against heat and pathogenic Vibrio challenge. Next, please. Now, this is a result which Irene, my PhD student, obtained just about, I think, last month or a few weeks ago, whereby when we knock down those heat shock proteins 70 in Nobli, in line with the heat shock protein 70 knockdown, and when she investigated the individuals of those Artemia, prophenol oxidase is also being reduced significantly. So there is a sort of like an indication where heat shock proteins is somehow involved in the prophenol oxidase pathway. But of course, this certainly required a lot more of research to look into the mechanistic action of prophenol oxidase activation in brine shrimp or even in Penis vanami. Next, please. And that brings out the question, where is the possible mechanistic pathway or mechanistic action of heat shock protein 70 in prophenol oxidase activation pathway. Next, please. All right, so these are the conclusion that we have, where we show that heat shock protein 70 plays a significant role in thermal and pathogenic bacteria tolerance in Artemia. And when we manipulate or when we upregulate those endogenous heat shock protein 70 expression, they can boost tolerance against heat and also against Vibrios. Next, please. Now, back in 2017, there is a paper from Chulalongkorn University which surfaced and indicate that when we give a non-lethal heat shock to Penis vanami, it has the same, almost the same effect towards Vibrio tolerance as what we have obtained with Artemia as a model organism. So this is very interesting, and we started to look into collaboration and see whether those tolerant is also linked with heat shock protein 70 and also other heat shock proteins. But because that we have developed the model of knocking down heat shock protein 70, so we have explored by using our method into looking at the expressions of this particular protein in vanami. So at Chulalongkorn University, they are doing big shrimps, juvenile and above. So juvenile shrimp might measure 3 cm and above. The thing is that when a shrimp is more susceptible to Vibrios or to white spot disease, it is usually during the post-larval stage, meaning that small shrimp, baby shrimps. So we tend to move into the idea of looking into the PL of vanami, and we started, I think, back in 5 years or 6 years, or even I don't have the exact time, but I would say 7 years ago, we started looking into knocking down heat shock protein 70 proteins, genes in Penis vanami. So next, please. So of course, other than tolerance against Vibrio, non-lethal heat shock also were shown to protect shrimp, white-leg shrimp against wide variety of stresses. Next, please. So we did a HSP knockdown with a minor modification of what we have done on Artemia. So if you look at here, this is a shrimp, a baby shrimp, shrimp PL post-larval stage, that we also deliver those double-stranded RNA. Same protocol, we construct the double-stranded RNA. Next, please. And of course, by constructing double-stranded RNA, we need to look into the heat shock cognate proteins of shrimp and also the heat shock proteins of shrimp. Next, please. And again, we construct the double-stranded RNA, but with Penis vanae, there is a slight difference in bringing in the methods of bringing down those heat shock proteins. And when we look at here, if you inject, next please, if you inject the shrimp with double-stranded RNA, and because they are they are slightly bigger than Artemia, then again the protocol of Artemia could not be totally exploited to this organism, and we have to do also an optimization how much of proteins, how much of double-stranded RNA that should be injected into this shrimp. So if you look at the video here, it is possible to inject double-stranded RNA into host larvae by micro-injection. And next, please. And this is how it looks like. Basically, you turn them into red color because of the dye, but the color will eventually fade off after 24 hours, just like what we observe in Artemia. Next, please. And when we optimize the knockdown, it is very interesting to see that if we inject 80 nanograms of double-stranded RNA into shrimp, if you inject only double-stranded RNA consisting only heat shock cognate protein 70 or heat shock protein 70, you might not bring down sufficiently the effect of knockdown. So you have to inject a combination of double-stranded RNA of these two heat shock proteins family protein into vanae, and with both combined, we could only bring down 71% of the expression and not the total, or I would say near to total obscuring of those proteins as what we have seen with Artemia. So the problem with knockdown in Penis vanae is that you cannot inject more than 80 nanogram with the available volume. And we, of course, we hope to inject more into it, but experimentally, it is not possible. All right. So the maximum that we can bring down until today, of course, there will still be optimization done, but it's only approximately bringing down of 70% of expression. Next, please. So we did an abiotic stress challenge on them, right? We imposed a pH and salinity stress on them, and we see that knockdown of those heat shock proteins significantly brought down also the tolerance against pH and salinity. This has already been published. Next, please. And next, please. And we see that based on the papers that has been published by Chulalongkorn University with the combination of the papers that we have produced, we could sort of like conclude that yes, HSP 70 plays a very important role in Vibrio tolerance. And again, manipulation by non-lethal heat shock can also boost tolerance against pathogenic bacteria. And I'm going to extend a little bit if time is still sufficient here, that I'd like to talk a little bit about the ways that we manipulate or the way that we use to induce heat shock protein 70 accumulation in Penis vanae. Next, please. So by looking into the way that we heat shock, of course, many of you will disagree to use heat as the only alternative of enhancing the expression of heat shock protein 70 in shrimp. Because if you do it incorrectly, then you might kill the animal, or you put the animal to extreme level of stress. So eventually, if you want to apply those technology in aquaculture, what we should do is to find a non-invasive way of stimulating the expression of heat shock proteins in aquatic organism. So we started looking at plant compounds that naturally induce heat shock protein accumulation in shrimp. And very nicely interesting also that we found one particular plant that consists large amount of what we call the heat shock protein 70 stimulating factor, or HSPs inducer, we call it. So there are compounds that is available in plants that might be used to induce HSP 70 accumulation in shrimp. And the plant that we found was, or is, next please. Yep, is a mangrove plant producing fruits. It's called Pandanus tectorius. A lot of that can be found in coastal areas of, I'm not sure in KL region, but then in Terengganu is certainly a lot that we can find along the coastal area. And we extracted the leaves, and also we extracted the fruits, and we extracted those compounds out, and we sort of like test them on the expression of heat shock proteins. Can you go up one slide, please? Sorry. One more slide up. Yeah. And if you look at the Western blot analysis on the bottom lower side, that you see that heat shock protein expression is highly expressed after exposure of 4 grams a liter in of those compounds to Penis vannamei. And along with that, with the exposure, you don't see that the compounds or any residues in those extract that could kill Penis vannamei, indicating that they are safe for application. And when we look at the right-hand side, the survival upon Vibrio parahemolyticus challenge, we see that the protection slowly goes up when you expose this shrimp to a very short-term bath just prior to Vibrio challenge. So we could use this extract to prime them, but of course there are still many studies to be done or to be performed in order to see what actually happens when this extract goes into the shrimp, whether they kill directly the Vibrio in their system, or in fact they have immune stimulatory efficiency by which relates to heat shock protein 70. So next, please. So we did some histology, and we found that those extract protected shrimp cells against the deleterious effect of Vibrios. Next, please. And we look into the hepatopancreas, and also we look into the muscles, where you see that those hepatopancreas and muscles, the damage done by Vibrios of those non-exposed shrimp is much higher than those that has been exposed to leaf or fruit extract, which consists of a lot of heat shock proteins induced. And for your information, heat shock protein 70 is largely involved in wound healing as well. So they promote a lot of wound healing. So, but those are all speculations and hypotheses. So we still need a lot of research to be conducted to prove that the hypothesis that we propose is true. And this serve as, I would say, the experiments of our lab in maybe 5 to 10 years to come. Next, please. And if you are interested, you can, of course, search for this topic, heat shock proteins and aquaculture and disease control in aquatic organisms. Of course, I do not have so much time to dwell into the results, but to give you a very brief and important results that we have found. Of course, if you like to know more, then you can always download our review articles that we published previously with our counterpart. Next, please. So a few people to thank here. Of course, my promoters, Professor Peter Bozier and also Professor Patrick Soglus, the late Professor Thomas McRae, and all my postgraduate students. Thank you very much for your attention. Thank you. So back to you, Prof. Subha.
Thank you, Yong. We will give 5 minutes for people to ask questions because after this, Yong needs to go for a meeting. So can we have the floor? Anyone want to ask question? Or you can put it in the chat if you don't want to speak. Anyone? Yes, Fikri. Go
Yes.
ahead.
Can you guys hear me?
Yes, Fikri.
All right.
Yes.
Thank you very much, Yong, for your talk. I just wonder why. How did you come to the? How did you find that the methanolic extract from that pandanus plant can be used to induce the expression of HSP70?
Yeah, thank you very much, Fikri. In fact, I have a good collaborator at our institute who has already conducted a lot of research using this plant. And for your information, at IMB, we work on natural products, and we have a lot of plants, material.
I see.
The collection that is available at the IMB, the Institute of Marine Biotechnology. And one of the things that we noticed is that a plant which usually grows in high salinity area, plants that belongs to mangrove or just beside the seaside or coastal area. And if you look at the literature, they consist a lot of heat shock proteins inducer in them, because they themselves require a lot of heat shock proteins, to withstand high salinity and high stress from the environment. So one of the reasons that we chose it is because that that is a plant that grow in high salinity area. And of course, there are some of basic studies that has been done by my colleague already. So this really gives us an advantage of going down a bit on our screening process, because this has been known already from
I can imagine you test a lot of plants, compounds from a lot of plants just to get identified. All right, thank you very much for your explanation.
Yeah.
Any more questions? No?
Can I have one more, just short one?
Sure, Fikri.
I wonder
Fikri,
how...
you can ask many more.
I can ask many more, no problem.
I just wonder, how do you immobilize the Artemia shrimp when you want to do the microinjection?
Yeah, that is a good question. In fact, we did not put any chemical to sort of like making them still. So it's quite easy that you just cool them
I
a bit,
see.
all right, and the agar is cool off, and when you inject, it don't move
I
too
see.
much.
Yeah, because
Yep.
you have to be very delicate to do that. I
Yes,
imagine
yes.
they wiggle around. It's such a nightmare. Thank you.
Yeah, and you need a lot of techniques,
Mm.
and if you ask me, I would not do it as good as my postgraduate students as well, because they are the one that really has the skill
The training.
to put them in. Yes, the training, and they do hundreds and thousands of animals.
All right. Thank you so much.
Yeah.
Any more questions? You have a lot of unusual people.
Sorry.
No, I think
it's
this
a
virus, right?
or something.
I think
Yeah.
virus type, I think.
Yeah. So please don't forget to fill in the evaluation form. If there's no more questions, Seba would like to thank you, Professor Yong, for giving us the time to present your work. And I think the young lecturers and also the young researchers and postgraduate students have benefited. We are really very intrigued with how you insert your siRNA into your double-stranded RNA into your Artemia. That was very nice. That's the skill. Is looking at the initial development of an animal is something that is very challenging, and we recognize your efforts, and thank you very much, Professor Yong.
Yeah, thank you very much, Prof. Suba. And of course, I welcome researchers from SEBA to collaborate with us if you are in the field, of course. And if any of you would like to play around with the RNAi on organism, you are most welcome to come here and join our lab.
All right, thank you. Thank you very much. Maybe if everyone can switch on their camera and we can take a photo.
Thank you very much.
Yep. Okay, who's taking the photo? Afiza, you need to take.
Okay.
All right. Okay, thanks, Yong.
Yeah,
Merry
thank you very
Christmas.
much. Yeah,
All right.
Merry
Happy
Christmas
New
and a
Year.
Happy New Year to all.
Yeah. Thank you so much.
Yeah. Thank you. Bye.
Bye. Thank you, Prof. Young and
Bye.
Prof. Suba.
Bye. Bye.
Thank you, everyone.
Bye.